In today's session of protein structure and engineering, we are going to take another topic of structure function relationship in protein in regards with hemoglobin. Okay, so here we are going to study the difference between normal hemoglobin and hemoglobin from a sickle cell anemic patient. So, the RBC of sickle cell anemic patient is morphologically changed in the shape, forming a farmer's sickle. So the sickle cell RBCs, as you can see in this diagram, they are not a disc shape, but they have formed a sickle or a curve shape, which will enable itself to get blocked in the capillaries, causing an anemic condition. So what generally happens here is the patient who are having sickle cell anemia will never suffer from malaria. Why? Because this malaria causing parasite plasmodium goes and gets its lodging in the RBC, takes the nutrient oxygen, produces its young ones as a trophozoite, burst open RBC, and it releases its trophozoites. But here, the structure of RBC is such that it doesn't find much nutrients in space, so the plasmodium will not infect the person suffering from sickle cell anemia. And you can see malarial cases in Africa and India. Okay, so what is the molecular basis of this sickle cell anemia? It is that the beta chain of hemoglobin on the sixth position, instead of glutamic acid, it is replaced by valine. So when valine replaces glutamic acid at sixth position of beta chain in hemoglobin, the structure of hemoglobin will change, like this, the structure of RBC will change from disc to a sickle shape. So this was studied by Linus Pauling using a electrophoretic mobility between the normal hemoglobin and a sickle cell hemoglobin. Later, this was confirmed by another scientist in 1957, was VM Ingram. So he performed a technique called as protein fingerprinting or peptide mapping, where the 2D structure of a protein is studied and compared and they found out that this is the molecular basis of sickle cell anemia. So this steps involve that you isolate the sample of hemoglobin from two different individuals. A normal hemoglobin and a sickle cell hemoglobin are taken in two different test tube, and it is treated with an enzyme called as trypsin. This trypsin will cut the protein into peptide at the residue having a amino acid called as arginine and lysine. So wherever in the protein it finds arginine and lysine, it will cut the peptide bond there. So the protein will now get converted into peptide. You take the sample and it is being spotted on a strip of Whatman filter paper. So what you take two strips of Whatman filter paper, and in each strip you're going to add one sample, and the sample is going to get subjected to a technique called as paper electrophoresis. So at acidic condition, these samples are going to run under the electrical field. So the highly charged peptide will migrate faster towards the opposite charge, cathode or anode. Okay. After the electrophoresis, the paper strip is taken, it is dried, and it is attached to the larger square of Whatman filter paper. And the next technique which we are going to follow is paper chromatography. So for paper chromatography, whatever separation is being done in paper electrophoresis, it's going to we're going to place that Whatman filter paper at the right angle to the electrophoretic direction, so that already in the previous technique the, the protein is separated into different peptide. Now you keep it in the direction where there will be a further separation of each of the peptide sample. So this paper chromatography is performed using a solvent, butanol is to water is to acetic acid in the proportion of 4 is to 5 is to 1. So in such condition, the separated peptide will move under the principle of partition coefficient between the solvent and the paper. So here what happens is, more the peptide is hydrophobic, it will move for a longer distance. So if the sample is more hydrophobic, it will travel the long distance. So after this paper chromatography is over, we take the paper, we dry it, we stain it with a reagent called as ninhydrin. If you remember, in 11th standard we studied to trace out protein, we use ninhydrin, which will give you Rumen's purple color. Exception is proline. In proline, it gives you yellow color. So here, when you trace out ninhydrin and when you compare both the strips, all the amino acids are same at the same location, at the same distance. Only difference would be, in normal hemoglobin, the glutamic acid will not be like will be present, which will be absent. The glutamic acid position will not be seen in sickle cell anemic sample, whereas there will be another amino acid traced, which is valent, which will be separated at certain distance. So here you can see this picture where the sample is here, like this sample is glutamic acid, which is in normal RBC. So this is the glutamic acid in normal RBC, which is being absent in the chromatographic paper of sickle cell RBC. So here it is absent, but instead of that, there is an additional amino acid distance, which is blue color, and that is valent. So now you can see the difference between this paper of normal RBC and this paper of sickle cell RBC. Okay, so whatever information we get it from protein fingerprinting, it is being saved into the database in the computer for the future studies of homology. Like if you take two different samples, you need to find the similarities and dissimilarities. You can compare it with the database of a computer. Now, the further more information in the database is added by another technique called as 2D electrophoresis, which was again called as O'Farrell technique. So this O'Farrell technique is 2D gel electrophoresis, where we use two different techniques. The first technique is isoelectric focusing, and the second technique is SDS page. So isoelectric focusing, here the protein or the peptide are separated based upon their isoelectric point. So now what is isoelectric point? Let us see the chemistry behind this technique. So isoelectric point is a pH. Actually, it's a pH at which the protein sample will not contain any net charge. Hence, once it has lost its charge, the migration of that protein will stop under the electrical field. So using this principle, a capillary tube is taken. On the capillary tube, we pour it with agarose gel, and on the agarose gel, a pH gradient is made using ampholyte. As you all know that this ampholyte are the molecules which contain both acid and base, which is used to make a gradient, pH gradient from 1 to 14. So after you make this pH gradient, you load your sample in the capillary tube, and the protein, depending upon its isoelectric point, it will migrate to the pH. Once it reaches that pH, its isoelectric point, the charge will become 0 and the protein will stop moving ahead and getting concentrated onto its particular pH or onto its specific isoelectric point. Now you need to take out the capillary tube after the separation and subject the same capillary tube onto a second technique called as SDS page. Now let us see what is SDS page. Generally, apart from agarose gel electrophoresis, which is used to separate DNA, there is another technique to separate the DNA, which is having a higher resolution, which is called as page. So polyacrylamide gel electrophoresis is a technique by which you separate the DNA based upon its molecular weight and charge. where you use acrylamide and bisacrylamide gel. The same technique is used here to separate the protein. But now, instead of a simple page, we run SDS page. Now, what is SDS? SDS stands for sodium dodecyl sulfate, which is an anionic detergent which gives a negative charge to the protein sample. Okay, so now you're preparing a gel which is made up of SDS page, sodium dodecyl sulfate, polyacrylamide gel electrophoresis. So you're going to make a gel using this, and then whatever peptide have separated on a capillary tube by using isoelectric focusing technique, you're going to lay that tube on one side or near the well of SDS page and allow it to run under the electrical field. And as I have told you, the SDS page, it works under the principle that lighter the weight, the more migration or the faster migration of the sample will be. So the sample will further get separated in the peptide series or lane, and once the electrophoresis is over, you're going to subject that page gel to a stain with silver stain, or you're going to add another stain called as Kumasi blue and compare the data of the sample and store the picture in a computer database for further analysis. So these two molecular technique are were very important, and in a exam point of view, they are asked for 5 marks. So protein fingerprinting and 2D gel electrophoresis we have seen today. So yes, please go through.